You're listening to the Knowledge Archives podcast. Welcome to the Knowledge Archives podcast. We're a group of students on a mission to learn from as many different disciplines of knowledge as possible. I'm your host, Madhav Malhotra, and today I'm glad to be joined by Dr. Jane Irwin, a lecturer at the University College Dublin in Ireland. Her research focuses on biochemistry and, in particular, microbiological functions with specific enzymes, which has allowed her to study many microorganisms, including very interesting species in the group of Extremophiles. These organisms have many incredible capabilities that can be helpful to humans, so I'm very excited to learn more about how her work led her to discover more about these fascinating species. I really appreciate you taking the time to talk a little bit about your research, and I'm looking forward to learning about this very intriguing area in microbiology. I thought, first of all, I would love to just ask you to quickly introduce yourself and explain a little bit more about how you got started with extremophiles and what some of your other research interests are.
Well, my name is Jane Irwin. I'm a lecturer in University College Dublin, so I'm Irish. And I got interested in extremophiles as part of a postdoctoral project that I did in the late 1990s. So my PhD is in biochemistry, and I have a particular interest in enzymes. So this particular project was funded by the European Union, and we were collaborating with a group in the University of Iceland. And the group was looking at enzymes from extremophilic bacteria that grow in the very cold waters off the coast of Iceland. So my job was to purify enzymes from some of these organisms and characterize them. And I did that and published some of that work. We were looking mostly at dehydrogenases, but we also looked at enzymes like proteases that can be used, for example, in processing fish. So this got me interested in the whole area of extremophiles. I went to conferences. I spoke to a lot of people who were working in this area. I also had a friend who was working in a related area. He was involved in finding biosignatures of life on Mars, and this linked with extremophiles. So I got very interested in the whole area, and I kept up with it by writing various reviews over the years. Now, my next job didn't involve extremophiles. I became a lecturer in a school of veterinary medicine, but I still retained this interest, and I was working in the area of glycobiology, characterizing enzymes that break down various different glycans, chains of sugars on cells, for example. But obviously, many extremophiles produce these type of enzymes as well too. So I suppose you are wondering, what do extremophiles do? What are extremophiles? So extremophiles are organisms that live under what we would regard as extreme conditions. These would be extreme relative to how mammals live, for example. So extremes of heat, extremes of cold, extremes of pH, extremes of radiation even. And the majority of these are bacteria or they're archaea, which are a particular type of unicellular organism, a third branch of life. So they're mostly unicellular, but not all unicellular. So for example, you could count the Antarctic ice fish, which lives in water at about -2 degrees as an extremophile. That doesn't actually need hemoglobin to actually transport oxygen around the body. Things like there's a little organism called the tardigrade, and that can survive between -272 and 151 degrees Celsius. So that's super tough. It'll live in vacuum, it'll live under high pressures. You can throw anything at it and it'll be fine. Cyanobacteria, some of these two, are also extremophiles. So there's all different types of extremes. I suppose the ones we might most readily think about are extremes of temperature. And that was what I was working on, what we call psychrophiles. So psychrophiles grow preferentially under 15 degrees Celsius, and they grow in the cold. You can find them at maybe -12 degrees Celsius. They'll grow, for example, in ice sheets, in little rivers of brine in the ice sheets. They'll grow underwater. They'll grow in cold water. So that's what I was working on. But I had colleagues who were working on enzymes from thermophiles. And thermophiles, these will grow between 60 and 80 degrees Celsius. And then you have a whole category called hyperthermophiles. And as the name suggests, these will grow at really, really high temperatures, between about 80 degrees Celsius. And the record is, I think, 129 degrees Celsius. There's an organism called Geogemi barossi, which will actually grow up to 129 degrees Celsius. So all these organisms are super, super tough. And then you get things that grow at extremes of acidity. They'll grow in really low pH, like the microbes that grow in the Rio Tinto in Spain. You'll get alkalophiles that will grow, for example, perhaps in very salty conditions, alkaline water, etc. These are very important too, for example, as sources of, say, laundry detergents. They produce enzymes that are very stable to alkaline conditions. And then you've got a whole category of what we call halophiles. So halo, this refers to salt. So these grow under conditions of high salinity. They'll grow somewhere between about 0.2 moles per litre all the way up to about 4 moles per litre, saturated salt solutions, 30% salt. So you find these sort of conditions in nature. You'll get them sometimes in places like the Dead Sea, for example. So many Israeli scientists work in this. They've got the Dead Sea literally on their doorstep. So they can get samples of various different types of halophilic organism. You get them in various types of salt pans. You get them in places like Spain. You get them in various parts of India. These are really rich sources of organisms that will grow in high salt. And then you have things that will grow under very high pressure. So at the very, very bottom of the ocean, and even at the very, very bottom down the Earth's crust, you'll find a category of microorganism that we call barophiles or pisophiles. And these will grow under high pressure. There are bacteria growing literally at the very, very bottom of the trenches of our oceans. So, for example, the Mariana Trench, that's under nearly 11 kilometers of water. And there are extremophiles growing down there. There are rocks thousands of meters beneath the surface. So, for example, places like gold mines in South Africa, they've found extremophiles. So these organisms have various types of adaptations that make them really, really resistant to various types of high pressure. Some organisms are anaerobic, so they'll grow in the absence of oxygen, and some are radioresistant. So there's a microorganism called Deinococcus radiodurans. This is so radioresistant, it will actually grow in nuclear power plants. It will survive under conditions of radiation that would kill other bacteria and would certainly kill mammals. You will find it, for example, in nuclear power plant wastewater, that under conditions that most things would die under. And then you'll find what we call xerophiles. These will grow under very, very dry conditions where there's very little water. And it's not just a situation where something is resistant to just one extreme. You get categories of microorganisms that are tolerant to multiple extremes. So, for example, you have thermoacidophiles, thermoalkalophiles. So there's an organism called Sulfolobus sulfitaricus, and this grows at pH 3, so it's acidophilic. It's quite happy at low pH, but it'll also grow very happy at 80 degrees Celsius. So a lot of these types of adaptations help each other. They help living creatures to survive under multiple types of extremes. It's the same with the bacteria living in the oceans. The lower depths of the oceans, it's about 4 degrees Celsius or less. So many of these organisms that are adapted to very high pressure are also adapted to the cold as well.
I think it's really amazing to hear about all of these examples of these different types of conditions that these microorganisms can resist, and to kind of think about how we've discovered all of these unique insights. I also wanted to talk a little bit about your perspective on this, because I know you were talking about how you became aware of extremophiles when you were working on your postdoctoral project. And then from there, you've been to conferences over the years and have seen some of the new discoveries that still kept you interested in this field. What are some of the ways that you've seen our knowledge of these kinds of organisms change over time?
I think we've learned over time how they're adapted. There's been a lot of work done over the last 20, 30 years exploring these at a molecular level. So, for example, what's the nature of the enzymes? How are the enzymes and the proteins in these organisms adapted to their conditions? So, for example, things that live in the cold, the psychrophiles, they've got very flexible proteins. The enzymes have active sites that are very flexible, very open. Whereas in order for organisms to survive under very high temperatures, the enzymes, the proteins have to be quite rigid. So a lot of work has been done on that. But another thing is too that science doesn't happen in a vacuum. Somebody has to pay for it. And a lot of applied work is done. So there's a lot of what we call bioprospecting, people gathering extremophiles from different environments, whether it's the bottom of the ocean or the Antarctic or the vents that we find at the bottom, say, of the Atlantic, the hydrothermal vents, that you've got whole colonies of different hyperthermophilic organisms living around them. People are really interested in looking to see if these have any interesting and useful compounds. For example, are they candidates for drugs? Do they produce compounds that might kill cancer cells? Do they produce compounds that might be novel antimicrobials in a world where we're running out of microbials? So I'm very interested in the adaptations, and I think more and more and more people are going to be looking at them as a source of very useful compounds that we can actually apply, particularly in medicine, because obviously that's really where the speciality is. But people are interested in them for a whole load of other reasons as well too. Another area too is bioremediation. So for example, some extremophiles can take up metals from the environment. So there are some extremophiles, they're often tolerant of acidic and alkaline conditions as well. And they can actually take up heavy metals. So if you've got an area, say an industrial area that's contaminated with heavy metals, in theory, you can seed it with extremophilic bacteria and they'll take up the metal and they might convert it to something else that's far less toxic. So I think that's going to be a big area in future in terms of cleanup, we'll say, of heavy metals, removing various toxic chemicals, also even radionuclides from the environment. They'll act as a kind of a cleanup operation, if you like. Another thing I was interested in too was the whole issue of they tell us about how life could exist elsewhere. We've only scratched the surface really of extremophiles. There may be literally hundreds, if not millions, of species of extremophiles out there to discover, and they're constantly being discovered. You might wonder, how do you find extremophiles? And there is a little bit of a problem with finding extremophiles sometimes, because they don't always grow very well in the lab. Some people describe it as the dark matter of microbiology. You know, if you want to grow E. coli in the lab, it's pretty easy. But if you want to grow something that only lives under very, very extreme circumstances, growing that is not easy. You need very specialized equipment, and sometimes we don't know how to grow them in the lab. So very often what they do now is bioprospecting. They'll get samples from a particular environment, and instead of trying to culture the microbes individually, what they do is they do what we call whole genome sequencing. So they extract the DNA, they sequence all the DNA, and then they match up the sequences of the DNA, particular genes within the DNA, not all the DNA, but there's something called the 16S ribosomal RNA gene that's used to catalog bacteria. And then they look at that, and they look at databases that have sequences of DNA for literally hundreds and thousands of organisms in them, and they use that to try and identify new species that are present. So that's a big area. Just trying to identify what's out there is a huge thing in itself. So that's another area that has come on in leaps and bounds over the last 10 to 20 years.
Mm-hmm. And we talk about the inherent challenges in trying to study these kinds of organisms and go out and one like bioprospect for them, but then also when we're trying to catalog them or trying to grow them in other conditions, we face a bunch of challenges. But obviously, it's very important given the uses that we can develop for these organisms. And you mentioned medicine, you also mentioned a bunch of areas like bioremediation that historically we've been using very human focused technologies, and now we're starting to look at how nature itself can help us with tackling these problems, which is kind of ironic given how oftentimes we're creating these problems. But it's very, very promising to consider the potential all this has. But when it comes to the future, like you were talking about, when it comes to these areas that you think will have more people giving attention to them in the future, what are some of the biggest questions that still need to be answered when it comes to extremophiles?
I think we've got multiple questions. We've got the specific questions. So, which organisms are present, for example, identifying them, cataloging them, and also finding ones that are useful from a human point of view. The microorganisms that make new drugs, new antimicrobials, ones that, for example, can be used for bioremediation. And we've really only scratched the surface. But there are broader pictures going on there too, because they can tell us something about the sort of extremes that life can tolerate. And this is where they have a relevance to the whole issue of astrobiology. And people are looking at extremophiles with a view to studying them to identify various different types of biosignatures that we can use to find life in habitable zones that exist beyond the Earth. So, for example, quite a lot of the studies now that are going on, people are looking at parts of the world that partly resemble places, for example, like Mars or Europa. Now, obviously, there's nowhere on Earth that's exactly the same as Mars, but there are places that have similarities. So, for example, in terms of dryness, in terms of cold, in terms of salinity, for example. So, one of the projects that a friend of mine is involved in, they've been looking at soils from the Atacama Desert. They're looking at biosignatures. They're doing chemistry to try and find signs of organic matter that would have been left there by extremophilic bacteria, and to see if you can identify ways of seeing where there would be microbes present if you got a similar type of soil from Mars, for example. And people are looking at this all over the world. So, for example, there are people looking at it in India. There's a place called Lunar Crater in Maharashtra in India. They're looking at it. They're looking at various types of saline systems in other parts of India. Places like the Rio Tinto in Spain, which is really, really acidic and full of sulfur. All these environments have similarities to environments that are outside the Earth. So people can study those as a proxy for extraterrestrial environments. So I think it's going to tell us something about life elsewhere in the solar system and possibly elsewhere in the universe. And we even know that there's bacteria well outside the Earth, because they've done studies with high altitude balloons, and they've found bacilli floating along above the Earth up to 40 kilometers above the Earth, which is quite extraordinary. And it also implies too that we have to be quite careful about spacecraft we send up, because if we do send spacecraft to other worlds, we have to make sure that we don't contaminate them, because some microbes can survive in the vacuum of outer space. They've been doing various experiments to see if they can survive, for example, outside the International Space Station. So we really have to be careful that we have everything properly disinfected before we send it off the world. So that's something that's of quite a lot of interest to me.
Mm-hmm. And I think it's so incredible to hear about all of these different areas of work, blending all of these different academic disciplines that you would never hear about with any typical organism, where you see that with extremophiles in particular, there are so many more possibilities that we don't even know about, that we haven't even begun to discover. So the last question I wanted to wrap up with was just to ask you, when it comes to one, discovering this knowledge, but then two, making use of this knowledge, do you think there are any key barriers that are getting in the way of this field growing more? For instance, in terms of the number of interdisciplinary researchers working in this field, in terms of the funding we have for these types of studies, or anything else in terms of the way we're going about conducting research instead of just the technical limitations we face?
I think there's quite a lot of money being invested in it, but it's being invested more towards the applied side of the science, the medical side of it, for example. Even things like the cosmetic industry, the food industry, these use it. So, for example, some of the sunscreen components actually come from radio-resistant algae. There's a compound called ectoin from some halophiles that's used in food and cosmetics. So I think part of the issue is there's plenty of applied research going on, but there probably isn't nearly as much funding available for the more blue skies type of research, the bioprospecting. So I think that we're going to probably make far more discoveries in the applied area than we will in the other area, because it's just the way that research funding operates. Funders want to get something back for their money. And for everything really, that's an issue.
Mm-hmm. Well, I think it will be incredible to see the research that comes in the future, even though that it might be exploring just a little bit of the incredible nature we have to discover about everything that still is unknown when it comes to extremophiles. But I really appreciate you taking the time to talk about these different disciplines and areas and kind of laying out the groundworks for why extremophiles are so important and incredible right now, but also how much potential they still have to teach us new things in the future.
Not at all. Thank you for inviting me. I really love this topic. I think it's a very exciting topic. And I think there's going to be a lot more discoveries coming out of it over the next few years. So thank you. It's a pleasure talking to you.